So I've covered each domain separately, so in this video I'm going to do a little comparison table of each of the 3 domains and some of the features. So to start off with, let's discuss whether they have a nuclear envelope or not. And the only domain where the members of it have a nuclear envelope is your eukarya. This is the same for if they have membrane-bound organelles. So only eukaryotes have membrane-bound organelles, your bacteria and your archaea don't. Whether they have a peptidoglycan cell wall is sort of the reverse. So only your bacteria have a peptidoglycan cell wall, your archaea and your eukarya don't. All of them can have membrane lipids. Both your bacteria and your eukarya have unbranched hydrocarbons, whereas your archaea can have some branched hydrocarbons. Regarding the RNA polymerases that are in these organisms, your bacteria only have one type of RNA polymerases, while your archaea and your eukarya can have several types. This similarity between archaea and eukarya is also seen in the amino acid indicator of protein synthesis. So in your archaea and your eukarya, they both use methionine, whereas in bacteria they use formylmethionine as the indicator amino acid. Something that's unique for each of the 3 domains is whether they have introns. So it's very rare for bacteria to have introns. Archaea can have introns in some genes, whereas eukarya often have introns in most genes, so it's very common in them. The next feature is just a bit of a nice one to know. So it's responses of each of the members of the domain to specific chemicals, such as your cerulophthalmicin and chloromethylphenolic. So usually this inhibits bacterial growth, but in archaea and eukarya, growth isn't inhibited. Another feature that's unique in between all 3 of them is regarding histones. So your bacteria have DNA that isn't associated with histones. Your archaea, they can have DNA that's associated with some histone-like proteins. And then in your eukarya, they are associated with histones. And then one thing that's unique for your archaea, because again, as you'll probably remember me mentioning, the extremophiles, so they can grow in places that are above 100 degrees Celsius, whereas your bacteria and your eukarya cannot really grow in such extreme temperatures.